Hey, welcome to Breakfast in Biology. This is day number 42.
Woo!
How many days we have? 11 after today.
Crazy.
It is crazy.
Who's in the house?
Oh, look, there's Nettie.
And Annie and Emily, Michael Jackson,
Maya.
Billy, where are you? Billie Jean. Maya's here. Darius, Mr. Noble is in the house. Jessica, Gladys, Cohen, Eli, Darius, Zuli, Dom. Who was first? Zuli was here. Eli's here. Carissa, Abby, Michaela, Lexi, etc., etc. Did we miss anybody?
I don't think so. So fun stuff today from your biology geek. Alexa, stop. Apparently she thought I said her name.
That's like when Siri all the time pops up. I'm like in class and I say something like, "Are you serious?" And Siri goes, "I didn't get that." Shush, shush, Siri.
Couple more. There's my lurker. Dominic says he was first. All right, so the fun. This is fun stuff for us today. I think we like this stuff. It's interesting. We're not going to give you anything to do afterwards. So today we're going to go over some kind of interesting genetic situations and share those with you and give you some insight into those. And then we'll leave, if you want to work outside of the live stream, we'll leave time for you to work on the stuff that maybe you still owe us. If you're all caught up, good for you. You get the rest of the day off, which is not the case for everybody. So please keep that in mind. Look at that stuff. Get it done. These are interesting. I think this is going to be interesting. You think it'll be interesting, Mrs. Smith?
Very.
Very. All right. Let's talk about some. And if we were in class, we would probably spend some time and do some of these problems and stuff. We're not going to give you any of these problems. Not today. We're not going to give you any of these problems. But there's some circumstances we want you to be aware of. So the other day when I was talking to you, we had a picture of Gregor Mendel up, and you guys had some interesting things. The 17th century Dwight Schrute, I think, was one of them. And you had some interesting ideas. And eventually we got to the fact that it was Gregor Mendel. And I said that he was like the luckiest guy in the world. Because the reason that we still talk about him in biology in 2020, the reason we still talk about him is because he started with pea plants and came up and did research, came up with the whole idea of Mendelian genetics. We named it after him. Had he started with the wrong organism, I don't think he would have got there. Or he wouldn't have got there as quickly. And so let us give you today some examples of genetics that maybe don't perfectly follow what he did, but we know about now. All right, so and what's really cool about this stuff is it all goes back to your ability to understand it, all goes back to the fact that you know that we have 2 chromosomes in every cell, one from your mom, one from your dad, matching pairs of chromosomes, homologous chromosomes. There's the fact that you know they replicate. All of that stuff leads you to be able to understand this stuff. So let's talk about the first one, incomplete dominance. I always thought this was pretty fascinating. Because for a long time, this is what people thought genetics was. If your dad was tall and your mom was short, then you were medium. And we know that's not necessarily the case. But there are organisms that have this idea of incomplete dominance, right? So neither allele is dominant. So far we've been talking about one of the chromosomes is dominant and maybe one could be recessive, right? When an organism is heterozygous for a trait, it will show a third phenotype. The third phenotype is a blend of the other two. In this example, letter A represents the gene. So gene is the A in this example is the G. So they're writing this a little bit differently, and the R and Y represent the different alleles. So we have an A big R, and A big Y, and A big Y. So look what happens. We have a red flower and a yellow flower. What do you need to take away from this? We have a red flower and a yellow flower. We put them together. If we get a heterozygote, we're going to get that flower in between. The classic example of that's typically like the red and white and pink, right? We have a red flower and a pink flower. And now they didn't write in this example the allele special, but they're telling you this is incomplete dominance. So we get a pink flower, right? So if you have a big R, if you have two big R's, you get red. If you have two little R's, you get white, but neither one of them is really dominant or recessive. And so sometimes the reason we would write it this way is because neither one is dominant or recessive, and we're used to writing big R's and little R's for dominant and recessive. So this is probably maybe better to help keep that straight. All right. Then there's another thing. We have incomplete dominance where there's a blending, but there's also something called codominance. So codominance is another type of dominance. Traits do not mix, but they both show. So we use capital letters for, well, that doesn't matter so much. Look at the example, and there's a better picture of it, I think, right here. We have a white cow and a red cow. And they, again, if you look, they're using big R's for the color, W's for white, little R's for red. When we mix those two, we end up with a roan color cow. Right? And so look at roan. It's white and red. They both show through. So incomplete dominance is a blending. Codominance, they both show. All right. So am I stalling enough for you, Mrs. Smith? Are you ready, or give you 1 second? Should I go back over it quick? Your mic's not on.
Oh, that's why. Okay. All right. I'm sorry.
Are you ready?
Yes.
Do you want to share your screen?
Yeah, I can do that. Sorry. Not on the right page. There we go. My children are also being loud in the background, so sorry about that.
They've become the background noise of our morning.
Yeah, no more peeing turtle. It's just my children. All right. So sometimes you have the situation where you have actually what we consider to be a multiple allele situation, where there's more than maybe two alleles that you're looking at. And one of the most common examples of this is blood type. And so if you think about the different blood types, you might know somebody that is type A, you might know somebody that's type O, you might know somebody that's type B, or you might even know somebody that is type AB. And now you might also be thinking in your head, oh, well, yeah, but somebody's O positive, O negative, etc. And I'll talk about that for a second, but that starts to get even more complicated. So the way that this works with blood type is that you have three kind of potential alleles. And the situations is that O is recessive to everything. So if you have type O, that means you have two O alleles. You have a very recessive blood type. A and B are co-dominant situations. So essentially, when you look at genotypes and blood typing, type O has two recessive O alleles. Type AB shows that co-dominance that Mr. Suter talked about, where you have one A and one B, and it's a co-dominant situation. But if you have the situation where you have type A blood, you can either have two A alleles, or you can have a type A allele and a type O allele, and then it will be type A because A is dominant over O. B, it's the same way. You can either have two B alleles, or you can have one B and one O. And so when we kind of look at this is just another chart that kind of shows you the same thing from the previous slide, where the genotype and the phenotype being the blood type, are a little bit different. You have different possibilities. And so essentially, when we talk about blood typing, there's only one type of O, there's only one type of AB, but you can have two different types of type B or type A. So what does all this blood typing mean anyway? Because we talk about like blood types, and just so you know, animals have blood types too. Like if you have a dog or a cat and you take them to the vet, and they've had their blood drawn before, they have their own type of blood, which is pretty cool. So I like this picture, and it does go a little bit above where we need to be. This is one I usually use in anatomy. But so what is type A? What is type B? What does that mean? So we've talked about proteins. we've talked about how there's proteins on cell membranes and how they can be used for recognition. And so essentially on a red blood cell, which is a fancy name for that is erythrocyte, which is why that's there. If you have type O, that means you have these proteins or antigens that grow that are a certain shape. If you have type B blood, it's because you have these antigens or proteins that are there that are produced that are type B proteins. If you have AB, both are shown. So you have both A and B. And if you have O, then you have the recessive and you have no proteins. So why is type O the universal donor? Type O is the universal donor because it's a raft without any proteins on it. So it can essentially fit into all of the different categories. So if you think about it like that, you know, in your body, if you have type B and all of a sudden I give you type A blood, your body, your antibodies, your fighters are going to recognize that as being foreign because there's something different on it. So they're going to attack it. If you're type B and I give you type AB, you have B on there, which they recognize, but they'll also recognize the A proteins that are on there. And so they'll attack that blood. But type B, there's no foreign proteins on it. So they'll accept it, which is why if you have type O blood, they love to get your blood because they can give it to anybody. The downfall is if you have type O blood, you cannot get blood from anybody else except type O. Because if we put any of the other ones with the proteins on it, your body will attack it. And so that's just kind of a quick overview. This is a really complicated chart, but it just kind of shows you if your father's blood type is A, B, AB, or O, what are the possibilities of their children depending upon the mother situation. So like I have type A blood. My husband has type A blood. So my children, dependent upon whether I am AA or AO, or my husband is A or AO, my children will either have A or O blood. Both my children happen to be A. It doesn't mean that I'm not AO. In fact, I know I'm AO because my father is O, but both of my children happen to get that probability. So this is like a kind of overly complicated look at that, but that is what's there. Okay, so when you're looking at blood types, it's both a regular traditional dominant situation where A is over O and B is over O and O is recessive. And then AB are the co-dominant situation where if you have one A and one B, both will be shown. The other thing we're going to talk about are sex-linked traits. So when we talk about sex-linked traits, I'm talking about the Y chromosome and the X chromosome. So if you think about females, they have 2 X chromosomes. But males have 1 X chromosome and 1 Y chromosome. And so when we're in class, I kind of like to joke a little bit about the Y chromosome because it's so tiny. And so, you know, when you deal with males, they really get most of their traits from the X gene that they got from their mom, from the X chromosome they got from their mom. The Y chromosome they had to get from their father. Their mother has 2 X's. They can't pass it down. And so they don't have really a dominant and recessive situation. They have a situation where they only deal with the X chromosome. Where females, they get an X from dad and an X from mom, so they become a female. But then they have that typical which one's dominant, which one's recessive, etc. So essentially, if you look at the X and the Y chromosome, the bottom half of the X and Y genes are similar, but the top, there's a much larger X section. And so there's not as many things on there. A lot of the things that are on the Y chromosome have to do with testosterone and the genes that make you a male. And so if we talk about hemophilia, which is one of the most common X-linked or sex-linked traits, is it is on the X chromosome. And so with sex-linked genes, there's a higher percentage of males that have these than females. Now, I want you to think about that for a second. Why would there be a higher percentage of males that have a sex-linked gene or sex-linked trait rather than females? Any thoughts to that? And as you're thinking, I'll answer Eli's question real quick. So O negative and O positive, it's actually another protein that's produced. So that's a typical dominant situation. So you are what we call Rh positive if you have that extra protein, and there's a long evolutionary thing we can talk about with that. Rh stands for rhesus, from the rhesus monkey, or negative means your body does not produce that protein. And so if you have positive blood, you can take negative blood. If you have negative blood, you can't have positive blood. And there's a lot of implications for women that have negative blood types and have children with men with positive blood types. They have to be given a shot so that their body doesn't attack the fetus. But good, Shay. Okay, so females have 2 X's and males only have 1 X. So if it's on the X chromosome, even if it's recessive, they have a higher chance of getting that particular thing. And so, and I'm talking a lot here, and I'm sorry, but as we talk about hemophilia, then you'll notice that the options for men are either you have it or you don't. So you have a normal male with normal blood cells, and then you have a hemophiliac male that has the 1 recessive gene. Where with a female, in order for them to have hemophilia, they must have both recessive genes. If they are heterozygous, meaning they have 1 of each, the dominant trait still takes over. So they are what we are called a carrier of something. Okay, so this is like a big Punnett square that kind of shows you, there's no alleles on the Y. And so you'll then have, 50% of this situation that the men will, or the sons will have the recessive trait. And for the females, even though mom's a carrier, none of the females will have the trait. So it's a little bit complicated. There's a lot of things that go into this, but we're just trying to give you kind of an overview with it. More with that, Mr. Suter, I need to say, or are we okay?
No, I think we're good. So just 2 other things. So I kind of one of the things we were thinking about here, what are some real world applications, right? So the blood type, real world application. So even though we're not getting as in depth as in this as we would if you were in school and you would have had to do that, and there were some Punnett squares you would have figured out for blood type and stuff, that's important for you to know, like to be just a citizen, right? And so what are some other real world applications of genetics? And these may not be important for you to know, but I think they're interesting. And so let me share my screen. Boom. One of them that comes to mind for me that I always thought was fascinating is in dogs. And the picture here is actually incorrect. I'm going to tell you why. So there's only 2 color labs. And I'm going to simplify this a little bit. I mean, we could probably get into a big argument, and Mrs. Smith could get into her high level AP biology or whatever. But just for sake of argument, there's 2 color labs. There's black labs and chocolate labs. And you go, no, Mr. Suter, you're wrong. There's yellow labs. I had a yellow lab, but she wasn't yellow. She was a chocolate lab. What are you talking about? Well, if you look at the genotypes that are on the screen here, look at the 2 black ones. It's big E, big E, big B, little b, big E, little e, big B, little b. So the B is for black, right? So the B is black. And if we get 2 recessive big Bs, we have a chocolate lab. Then we come over here and we see the yellow labs, and the only difference between them is they just have this little E here. So what does that mean? And this picture is actually incorrect. Why? Because, you know what? I'm going to skip over that one. Look at these pups right here. In that picture, I see 3 black labs and 2 chocolate labs. And you go, no. Look at their noses. Black lab, black lab, black lab, chocolate lab, chocolate lab. So what is happening here, and we told you in the beginning, is sometimes there's more than one gene that controls things, right? There's one gene that controls black or brown, but there's another gene that controls whether that color gets into the hair. So in that big E that we saw in that last slide, where lowercase e's, it's turning off whatever mechanism or whatever allows that color to get into the hair of the dog. That doesn't stop that color from getting into their lips and their nose. And so that's really interesting because you can see now the yellow lab that I used to have, her name was Susie. I don't think I have a picture of her right here, but I had the yellow lab named Susie. There was a picture of her on my desk, I think. She was a chocolate lab with yellow fur because the color wasn't into the fur, right? And so here's another thing to think about. The breeder actually told me about that. But another thing to think about is genetic diversity and stuff. And so when you are going to buy a pet, labs became very popular at one time. And breeders started breeding them very heavily. And you get like puppy mills that are breeding dogs. And so when you're buying like a purebred dog and you get it from a puppy mill, one of the problems you run into is a lot of inbreeding. And if you have a fully recessive gene, think about every Punnett square we've done. When you get those fully recessives, so a yellow lab with a brown nose is totally recessive for the color and totally recessive for whether it goes in the skin. To get a lot of those, they were sometimes pretty heavily inbred, and you can find that you have problems with dogs like that. Sometimes. Actually, and she had a lot of problems with her hips and stuff as she got older. I don't know that it was because of that, but just something to think about, right? Let's move on. In the back of the room, we have a corn snake. That's not this corn snake. We used to have this corn snake up until a year or so ago. Well, not this particular one, but this color. This is the normal color corn snake. Corn snakes are interesting because they have two genes that control the color, right? So one gene controls the brown color. And if you take that brown color away, the melanin away, we get what's called an amelanistic snake. And that's the snake that would be like the one in the back of the room, right? That orange one in the back of the room. It's missing. It's recessive for melanin. It's amelanistic. Another variation takes away the orange color. That's called antheristic. So this would be an antheristic corn snake. We've taken away the orange color and it's all brown. Another option would be to get recessive for both of those, take both of those colors away, and we get what's called a snow corn. And then if you go and you type designer corn snakes into Google and look it up, there's striped ones. I mean, people have messed with their genetics in captivity. There's all different colors. There's hypomelanistic, hypoantheristic, like really bright oranges, really bright, just all kinds of cool stuff happening. And so here's what that looks like. And this is, I just want to show it to you quick. We're not going to dive into it, but if we kept going with Punnett squares, once you start adding multiple genes, so big B, little b, whoops, big B, little b, big R, little r, the B is for black, the R is for red. So if you have two little B's, you're missing the black, two little R's, you're missing the red. You can see we could do a giant Punnett square. And what's interesting is if you do two heterozygotes, we do two heterozygotes on a monohybrid cross like we've been doing, we get a 3-to-1 ratio. If you do it with a dihybrid cross, you get a 9-to-3-to-1 ratio. So we have 9 normals, potentially, 3 blacks, potentially, 3 reds, potentially, and 1 snow corn. So again, to get to that snow corn, that's a rarity. And you find the same thing in the animal with breeders in the animal trade. Whenever you're looking at an organism that is exhibiting all the rarest like recessive traits in the pet trade, sometimes they've been heavily bred for that. And it's just something to be kind of think about. Anyway, I think Mrs. Smith, if you want to jump back in with me, the last thing that we had was just kind of this thing, which we thought was kind of cool, right? Showing the difference between complete dominance, codominance that we talked about at the very beginning, and incomplete dominance.
And don't get overwhelmed. Like you saw some of the genotypes and blood type here. And when we were talking about the sex-linked genes, where it's like the exponent. That's just how it's written. You know what I mean? If you decide to go AP route, we'll use that because there's reasons for it. But don't get overwhelmed. Like what we're looking at is not the C here. We're talking about the letters that are up there. So you can see the genotype where it's big B for the first one, The second one is big Y, and then the third one is the B and the Y.
All right. So again, we dumped days worth of talking to you in practice and looking at it on you because we want you to be aware that it's out there. So sometimes in education and in learning, it's better just, it's one of these things. If you don't know, you don't know, right? So if you're not sure about AP Bio in the future, you're not sure about whatever, we wanted to let you see there is more to the genetics than just doing a Punnett square, right? And we're just scratching the surface. So you can go to college and you can major in bio and you'll take a whole genetics class that will go deeper, or you could focus on genetics and take lots of genetics classes and become a geneticist and really go deeper. So there's a lot more to this. It's still the classes that you're taking now as a freshman or a sophomore are a really wide look at everything that's happening. You could pick a piece of that and go, wow, I really like ecology, or I really like evolution, or I really like genetics, or I really like organic chemistry. Right? And you could dive into that and make that a career and learn more about that. So remember, as you're going through high school, it's the exposure to the stuff that maybe will trigger something where you go, wow, this is really cool. I want to know more. So anything else, Mrs. Smith?
No, I really like genetics. So if you have any questions, and I hated it when I first took my first genetics class in college, because it is overwhelming. There's a lot of stuff there. But if you start thinking about, how it all works together and the big picture and the fact, of how we look and everything has to do with teeny little things inside of individual cells, and that causes your body, if you think about that whole transcription to translation process of making proteins, starts to get pretty cool. Why do I have freckles?
Yeah. And when we were in college, it was still kind of the old days. Those old geneticists used to play with flies a lot, and they used to put the flies to sleep with something called, I think, with ether, right? And then they spent a lot of time around ether. So every genetics professor I ever met in college was a little bit lightheaded.
Yeah, we use fly nap now. It's a little different.
They were a little bit lightheaded. Spent too much time around the ether. All right. If there's nothing else, please, can we get everything made up and caught up? You feeling that way, Mrs. Smith?
Feeling it.
Yeah, just get it done. And we'll see you tomorrow, I think. So with that, what do we say? We miss you. We love you. Wash your hands. Stay safe. And stay away from the ether. See ya.